My name is Marissa, and I will be presenting on how metformin can be used as an intervention to attenuate the negative effects caused by aging. Metformin is an oral diabetes medication. It is commonly used to treat type 2 diabetes. It does so by lowering blood glucose, cholesterol, and triglyceride levels. Metformin decreases peripheral insulin resistance and decreases intestinal glucose absorption. Diabetes is an independent risk factor for cardiovascular disease development. This is important because, as discussed in class, cardiovascular disease is one of the leading causes of mortality and morbidity in humans. Metformin is protective beyond the hypoglycemic effect. As shown on the figure in the left, metformin has a number of beneficial effects. A few of these are increased heart function and inhibition of NF-κB. NF-κB is a pro-inflammatory transcription factor. This means that if you inhibit NF-κB, this leads to the prevention of oxidative stress. As a quick refresh, increased oxidative damage leads to cellular senescence. This can be described using the metaphor of a sick patient that has a virus. This patient is alive but is not functioning because they don't feel well, and this patient can infect nearby people. This is very similar to a cellular senescent cell because this cell is alive but it's not functioning, and it infects nearby cells via inflammatory signals. Lastly, senescent cells are very difficult to kill, and this is what makes them so dangerous. Now that we know metformin has a number of beneficial effects, we can look at what previous studies have tested. Previous studies suggest that metformin increases longevity. This is very important because this means that metformin may be used as a therapeutic use to prevent cardiovascular disease in individuals with and without type 2 diabetes. Now, I can ask my big question. How does metformin supplementation affect aging mice that are otherwise healthy? The study I chose to read is called The Protective Effect of Metformin on D-galactose-Induced Aging Model in Mice by Fatemi et al. In 2018. This was a randomized control study consisting of 50 male mice. There were 4 experimental groups and 1 control group. Of the experimental groups, there was 1 group given only metformin, 1 group given only D-galactose, and the other 2 groups I have shown down below. Both of these groups were given D-galactose, with 1 group given the low dosage of metformin and the other given the high dosage of metformin. These various dosages are the reason that I chose this article. I thought it would be really interesting to determine if varying dosages of metformin would have differing effects in their attenuation of D-galactose-induced aging. To give you all some quick background on D-galactose, D-galactose induces aging, meaning that it causes effects that are often seen by aging individuals. These include increased oxidative damage, memory impairment, and reduced physical endurance. The big question asked by this article was, how does supplementation of metformin affect age-related processes in general? And more specifically, how does metformin attenuate the negative effects caused by D-galactose-induced aging in mice in factors such as anxiety, oxidative damage, memory impairment, and physical endurance? Lastly, how does the dosage size of metformin alter its attenuation of these negative effects? Here, I have attached a figure from the study. A novel object recognition test was completed in order to determine the discrimination ratio, meaning their preference for a novel object. This is commonly used to determine memory efficiency. Here on the y-axis, we have working memory, and on the x, we have the groups that were studied. The number sign on top of the group provided only DGAL indicates that this group has significantly impaired memory function in comparison to the control group. Furthermore, the group provided the larger dosage of metformin in addition to DGAL displayed a significant improvement in memory function in comparison to the group supplemented with only DGAL. While the group supplemented with the larger dosage of metformin did display these attenuating effects, the group given the lower dosage of metformin did not have this significant improvement in memory function. While this group given the lower dosage of metformin does seem to appear to have improved memory function in comparison to the DGAL group, there was no significant difference found between these 2 groups. This also answers the big and specific questions of the study because it shows that a larger metformin dosage is able to attenuate the negative effects caused by D-galactose, specifically working memory. Here are the key results. As shown on the previous slide, the group given the larger metformin supplementation does improve working memory, but this is not seen in the group given the smaller metformin supplementation. Furthermore, there was increased SOD superoxide dismutase and BDNF brain-derived neurotropic factor proteins in the larger metformin supplementation group. These proteins function to reduce oxidative stress. This means that larger abundances of these proteins means that they are able to reduce more oxidative stress. Less of this was seen in the smaller metformin supplementation group because only increased SOD, superoxide dismutase protein, was recorded. Although this is true, both groups were able to prevent weight loss, increase physical endurance, and reduce anxiety. This means that the big question was answered because metformin was able to attenuate negative effects caused by D-galactose-induced aging. Also, this answers specific questions because a larger metformin supplementation group was shown to be more beneficial than the smaller metformin supplementation groups. The specific factors recorded are also shown here on the Venn diagram. The main conclusion found in this study is that larger doses of metformin have the ability to attenuate negative effects caused by D-galactose-induced aging, including increased anxiety and oxidative damage, memory impairment, and reduced physical endurance. Some implications from this study are that metformin is readily available. As I discussed earlier in this presentation, metformin is already prescribed to patients who have type 2 diabetes. This means that if it is found that metformin can be used to prevent human age-related conditions in the future, metformin would be much easier to prescribe than any newly created medications. Lastly, this type of research leads to more research studies regarding aging preventions. The limitations that I found in this experiment were pretty relevant to what was discussed in class. First, this experiment was completed in the D-galactose-induced model of aging. It should be really completed in an actually aged model of mice in the future. Next, only male mice were studied, and as discussed in class, this has been a huge issue in research because males and females should be studied. Lastly, mice were used as subjects, and of course this has to be used first. In the future, humans will need to be used for experimentation purposes. This experiment should be repeated in an aged model of female and male mice, and eventually should be completed in humans. Thank you so much for watching!